This actually isn't a very difficult lecture. There's a little bit of math we're going to need to work through. Mostly it's conceptual, and there's some memorization with regards to naming conventions, but it's not too bad. Your goals for this lecture are to understand and describe the structure of the mitochondria, pretty much straightforward. Then we're going to calculate free energy and potential ATP generation using a redox pair and the Faraday equation, and that sounds more complicated than it actually is. Then we will name the electron transport complexes and describe the flow of electrons through the system for ATP generation. So mitochondria, most people have sort of a generic concept of what they look like, but in reality, mitochondria do have different shapes and they can appear in different numbers depending upon the particular cell type you're examining, but they all have sort of a common overall structure. There's a double membrane system, first of all. So we have an outer membrane and an inner membrane. The outer membrane is somewhat leaky by comparison. You'll understand what this means fairly soon. The inner membrane is essentially impermeable. It is comprised of a large number of proteins as well as phospholipids. Its percentage leans towards protein over phospholipid. It is essentially impermeable, and it needs to be because the space between the membranes contains protons, and a lot of them. So the inner membrane is basically setting up a proton gradient. So we have a huge concentration of protons on the outside of the membrane and basically very few protons on the interior portion or the matrix portion of the membrane. And that brings us to the matrix. The matrix is the center chamber, if you will, of the mitochondria. It houses reactions of the TCA cycle, it houses reactions of heme biosynthesis, it houses reactions of the urea cycle, it houses reactions of beta oxidation, and so on. And so as you can see, the mitochondria are far more important than simply making ATP. They do a large number of activities. Then we have these other structures called cristae, and these are inward folds of the inner membrane that increase the surface area of the inner membrane. And this is kind of what gives mitochondria their signature appearance. So here's just a couple of electron micrographs just to show you what they look like. We have mitochondria from hepatocytes, rat liver mitochondria, and then rabbit heart or cardiac muscle mitochondria. And you can see they are shaped rather differently, but we can still see these inward folds. These are the cristae here, and there's a lot of them here. So we can see that sort of inward folding, that almost maze-like or labyrinthine appearance, is very common in mitochondria, despite whether or not their shapes and numbers and overall structure changes. So as I've kind of already alluded to, the outer membrane of the mitochondria is somewhat leaky. It's 60 to 70% protein, and there are some porins, voltage-dependent porins, that allow particles of about 10 kDa in size to pass. The inner membrane, however, is a lot more complex. As you can see, it's almost 80% protein, and there's all kinds of dehydrogenases and transport systems, mostly acting on that proton gradient that I mentioned earlier. So here's just sort of a generic mitochondrion. We have the outer membrane here. We have the inner membrane shown here. We have these cristae, which are the inward folds. And then studying the surface, really kind of spanning the membrane from the matrix to the inner membrane space, are the complexes of the electron transport chain. So that's a mitochondria. In the matrix, as I said, we have TCA enzymes, fatty acid oxidation or beta oxidation enzymes, porphyrin synthesis related enzymes, urea synthesis enzymes. There's also, mitochondria have their own DNA, and they contain ribosomes. So they actually do create some of their own proteins. Not all of them, but a significant number of the mitochondrial proteins are actually made in the mitochondria. And so what are we seeing overall with regards to energy generation is we have, let's just suppose TCA cycles in the middle, and the TCA cycle is generating electrons as well as some CO2, but the CO2 is a waste product. The electrons are going to be fed into the electron transport chain, and while this diagram is showing only one complex, there's actually a whole series of complexes. There are 4 complexes, and some of those complexes, most of them, are going to be pumping protons out of the mitochondria matrix and into the inner membrane space. And so we can see we have our voltage differential here, as well as a chemical differential. So we have an electrochemical gradient being formed between the inner membrane space, the outer membrane is not shown, and the matrix. Oxygen is required to be an electron acceptor. It will become water in the process. And by doing this, we have this huge electrochemical gradient, which then another complex called the F1F0 ATP synthase can allow protons to flow back through, and using the energy of that motion, of that electrochemical gradient, the F1F0 ATP synthase is going to combine ADP with inorganic phosphate to become ATP. So this is sort of the overview of how the electron transport chain works. You have the TCA cycle making reducing equivalents, including NADH and FADH2. Those reducing equivalents are going to enter into the electron transport chain. The electron transport chain is going to move electrons down a redox gradient, and in so doing allow protons to flow from the matrix to the inner membrane space. And then this is driven by the reduction of oxygen to water. By moving protons in this way, we get this electrochemical gradient, this voltage gradient, along with the proton gradient. And so that's the overview. So now we're going to move to how the electron transport chain works. What is the theory behind it? We'll get to the complexes in a moment. So first, what we're going to be looking at is the oxidation of NADH and FADH2 as they're going to move electrons from NADH, that's oxidation, onto a carrier or onto transport mechanisms and ultimately onto oxygen. We have to have oxygen in order to draw the entire process forward. So theoretically, complete oxidation of a mole of NADH or FADH2 by the electron transport chain results in about 2.5 moles of ATP. It's a little bit less for FADH2, it's only 1.5, as you'll see why in a moment. So essentially, we have linked up a series of oxidation reduction reactions in order to capture the energy. And one of the reasons it's linked up stepwise is we lose less energy to heat. Okay, so if we did this in just one jump, just FADH2 or NADH to oxygen, we would actually lose more energy to heat due to the second law of thermodynamics. So instead, we're going to move things down oxidation reduction steps. So just some vocabulary. Oxidants are compounds that readily remove electrons from other compounds. In other words, they oxidize other compounds. Oxidants oxidize by removing electrons. Reductants are compounds that readily give up electrons to other compounds. So reductants reduce other compounds. Now, an oxidant and a reductant are always paired. In order to get this electron transfer in this way, we are literally pairing an oxidant with a reductant. So in other words, oxidants remove electrons from reductants. By definition, the material that is having the electrons removed from it is a reductant because it is placing electrons onto another material. The material that is doing the removing is by definition an oxidant because it is drawing electrons away from a material. So by definition, oxidants are reduced and reductants are oxidized, and they are found as redox pairs. So if we just look at a simple example here, we have cytochrome C iron 2 plus and cytochrome A iron 3 plus results in cytochrome C iron 3 plus and cytochrome A iron 2 plus. We clearly had an electron transfer because we go from iron 2 plus to iron 3 plus and iron 3 plus to iron 2 plus. So an electron was moved. So cytochrome C loses an electron. It goes from 2 plus to 3 plus. Remember, electrons are negatively charged particles, so we lost one negative charge. And cytochrome A's iron gained an electron because it goes from 3 plus to 2 plus, so it must have gained a negative particle. So we can always ask the question, what was oxidized and what was reduced? Well, we know the reduction is a gain of electrons, but more importantly, we can just look at the number. So it's called reduction because the charge was reduced. It went from plus 3 to plus 2. Or if we went from minus 1 to minus 2, that'd be the same thing. We're increasing, if you will, the negative of the charge. It's being reduced. It's getting lowered. Oxidized is the opposite side of this, where a charge is going to increase. So we go from plus 2 to plus 3. That indicates a loss of an electron. Loss of electrons is oxidation. Gain of electrons is reduction. So what was the reductant and what was the oxidant? Well, cytochrome C was the reductant because reductants are what do the reducing. So the reductant must give up its electron to the oxidant. The oxidant must take its electron from the reductant. So cytochrome C is the reductant. Cytochrome A is the oxidant. Cytochrome A oxidized cytochrome C, and cytochrome C reduced cytochrome A. Work with this equation until you're really, really comfortable with juggling these terms. Now, often we see electrons are being transferred with a proton. So for example, we might see something like this, where NADH plus proton plus FADH results, FAD, that should be FAD, results in NAD plus FADH2. So here, what we have is NAD giving up electrons onto FAD, and then we see the charge on NAD goes from 0 to plus 1. We don't really see the charge loss on FAD. It should be FAD plus, and FAD plus becomes FADH2. We can ask the same question, so I'll leave this to you to work out what was oxidized, what was reduced, what was a reductant, what was the oxidant, and why, and make sure you can explain that entire story. So now we have this idea that certain materials will give up electrons to other materials. We call those materials reductants. We call the materials that are accepting the electrons oxidants. Reductants are oxidized, oxidants are reduced. But what is going to determine whether or not something is going to act as an oxidant or a reductant within a redox pair? It's almost similar in idea that when we look at electronegativity, we know that an atom is more likely to accept electrons than some other atom. For example, chlorine is more likely to accept electrons than lithium. We can do this similarly with the electromotive force. Now, we're not looking at particular atoms here. It's not electronegativity, but we are looking at the electromotive force of compounds. And so the electromotive force is going to have one of two values. The electromotive force can be positive, which means the reaction is going to tend towards the right, or electromotive force can be negative, which means the reaction tends to the left. And so I'm going to show you what that means. So strong reducing agents want to give up electrons. They are going to have a negative electromotive force value, and the reaction will tend to proceed to the left. So oxidizing agents, on the other hand, want to accept electrons. They're going to have a positive electromotive force, and the reaction will tend to proceed to the right. But whether or not something's going to be an oxidant or a reductant is going to depend upon the context of the specific redox pair. So let's look at two oxidation-reduction reactions. Now, one thing to notice is every time an oxidation-reduction half reaction is written, it is always written with the oxidized form accepting electrons to become the reduced form. So we have acetate with electrons becomes acetaldehyde, or oxygen gaining electrons becomes water. And this is going to have some sort of voltage. So for example, the acetate to acetaldehyde, where acetate gains electrons, actually has an electromotive force of negative 0.60. And what that negative tells you is that generally acetaldehyde wants to give up electrons. This reaction wants to run to the left. Whereas look at water. Water has an electromotive force of positive 0.82, which means generally oxygen wants to gain electrons to become water. Water really doesn't want to give up electrons, which we know water really isn't a reductant, or, on the other hand, oxygen, think of the name, oxygen is absolutely an oxidant. So we can look at this and determine the stronger oxidant, okay, the one that wants to pick up electrons, the one that wants to become reduced, has the most positive value, whereas the stronger reductant has the most negative value. Now, that does not mean that the reductant will necessarily have a negative value. It may, but remember, this has to do with context of the redox pair. It is simply the most negative is the reductant, the most positive is the oxidant. So electrons are going to flow from strong reducing agents to less strong reducing agents to oxidizing agents, and they will not flow backwards in the electron transport chain. So what we're going to see in the electron transport chain is that electron carriers are going to offload their electrons onto their cognate complexes. So NADH will offload electrons onto the NADH ubiquinone oxidoreductase. FADH2 is going to offload electrons. You'll see it doesn't actually offload electrons to succinate dehydrogenase, because that is the complex that picks up the electrons, but will offload its electrons onto ubiquinone. So then the electrons will move from ubiquinone to cytochrome C reductase, onto cytochrome C, and then onto cytochrome C oxidase, and then on oxygen. The electrons do not run backwards in this system. Unlike many enzyme-driven reactions, we don't actually see backwards flow here due to the energetics. So when we look at standard reduction potentials, not that you would memorize this chart, you'll see we have those same sorts of reactions that we saw previously. So for example, here's acetaldehyde as an oxidant to ethanol, and we get movement of 2 electrons from acetaldehyde, onto acetaldehyde to become ethanol, and that has a voltage of negative 0.2, whereas oxygen will pick up 2 electrons and become water with a voltage of plus 0.82. And so we can see that all of the reactions are written in the same direction. Oxidant plus electrons yields reductant, and the idea here is to, when we see a redox pair, is determine which one is going to be the reductant and which one is going to be the oxidant, because we need that information to solve this equation. So we can figure out the relationship of free energy to the change in voltage of a redox pair, and that is given by the Faraday equation. So we have delta G naught prime is equal to, and please notice there is a negative sign in front of this expression, the number of electrons transferred, the Faraday constant, which is basically 23.06 kcals per volt molar, and then the change in the electromotive force, the change. And I'm going to show you how to calculate that. The key to solving this equation is determining which material will be a reductant, which material will be an oxidant, and then realizing you can use this equation to calculate a delta G. And once we have a delta G, we can calculate a theoretical creation of ATP. So let's look at an example. So we can have NADH plus oxygen plus ADP plus inorganic phosphate is ultimately going to result in NAD plus water plus ATP. So we go from NADH to the formation of ATP. So we're interested in how much energy is going to be available in the redox pair of NADH and oxygen, so that we can use that energy to create ATP. I'm not claiming this is a balanced equation. It obviously isn't, but I'm showing you an overall concept. So if we look at the direction of electron flow, we have NAD is going to become NADH plus proton. This requires the transfer of 2 electrons and has a voltage change of negative 0.32, or voltage of negative 0.32. Oxygen, on the other hand, is going to pick up 2 electrons and become water, and that has an electron change, an electron voltage of 0.82. So the difference between these voltages is the actual delta E that we're looking for. However, it is not sufficient to simply add up these voltages. You might be inclined to say, well, negative 0.3 plus positive 0.82 is positive 0.5. That would be wrong. Because remember, we actually have to track electron flow. And right now, as written, we are not, because both species are gaining electrons. We need one of these species to lose the electrons so that the other species can gain it. We need the reductant to lose electrons to the oxidant. So you can probably guess where this is going. So what we need is that electron potential difference, and that difference is going to drive the electron transport chain. So let's look at it. This is the same reactions just taken from our table. Again, I would not ask you to memorize those reactions or values. That would be silly. But you can see our oxygen gaining electrons positive 0.82 volts, and our NAD gaining electrons to NADH is negative 0.32 volts. However, what we really need is the electron flow. So what I've done in this slide is I reversed the NADH equation. You'll notice now I have NADH loses 2 electrons to gain, or to become, NAD. And because I reversed the equation, I have to flip the sign. So now we have NADH losing electrons, oxygen gains those electrons to become water, and we have a flow of electrons. NADH, the electrons come off, then they go on to oxygen, and we have from NADH becoming NAD, it is oxidized, oxygen gaining those electrons to become water, it is reduced. Now we add up our voltages, but notice the sign has changed, and instead of getting 0.5, which is what we might have anticipated based on the negative 0.32, instead we get 1.14. All right, so now that we have 1.14, we also know how many electrons were transferred, 2. We know the Faraday constant is about 23. We can actually put all of that into the Faraday equation. Do remember there is a negative sign sitting in front of all of this. So we have our delta G naught prime is equal to negative 2. That's the electrons that were transferred, number of electrons. Our Faraday constant, 23. And our delta E of 1.14, this gives you negative 53 kcals per mole energy. How much ATP can that make? Well, theoretically, we would use a value of ATP based on hydrolysis of 7.3 kcals per mole. So 53 divided by 7.3 is probably, theoretically it'd be 7.82 ATP per NADH. However, we already know that the second law of thermodynamics means we do not get perfect energy conversion. So some of this energy is lost as heat, which is not useful for making ATP. So we will not get 7.2 ATP per NADH. We're going to get something less than that. All right, so with that out of the way, let's now lastly look at the complexes of the electron transport chain. So how does this all work? There are 5 complexes in the electron transport chain that are used to make ATP. Now, one of those complexes does not move electrons at all, and I don't mean it moves protons back into the matrix, so it's not really an electron transporter. As the electrons are moved from reducing agent to oxidizing agent, we're going to make the proton electrochemical gradient. So overall, here's how it works. Complex 1 is going to use as a substrate NADH. Its name is NADH ubiquinone oxidoreductase, so it's going to oxidize NADH and reduce ubiquinone. That's complex 1. Complex 2 is succinate dehydrogenase, which is literally the enzyme from the TCA cycle, by the way. And this might also be called succinate ubiquinone oxidoreductase, but it's going to use FAD as a prosthetic group, and FAD is going to pick up electrons to become FADH, and then those electrons will be transferred onto ubiquinone. So as I said, complex 2 doesn't really take in FADH. FADH is part of complex 2. Complex 2 is actually a TCA cycle enzyme. So both complex 1 and complex 2 are going to offload their electrons onto ubiquinone, which will become ubiquinol, and then ubiquinol will offload its electrons to complex 3. Complex 3 is formally known as the ubiquinol cytochrome C oxidoreductase, or sometimes the cytochrome BC1 complex. Now that's complex 3. So we have complex 1, NADH, complex 2, FADH2, then we have ubiquinone, not really a complex, then complex 3. Complex 3, the cytochrome C oxidoreductase, is going to put those electrons onto cytochrome C. Cytochrome C is located in the intermembrane space between the two membranes. The rest of these are integral membrane proteins. And then cytochrome C will pick up the electrons from complex 3 and deposit those electrons onto complex 4. Cytochrome C literally migrates across the membrane to complex 4. Complex 4 is also known as the cytochrome C oxidase, and it will collect electrons from cytochrome C to put them onto oxygen. Now, really quickly, complexes 1, 3, and 4 all pump protons. Complex 2 does not. None of these complexes make ATP. So complex 1, the NADH ubiquinone oxidoreductase, we're going to transfer electrons from NADH to ubiquinone, sometimes called coenzyme Q, you might see that. Complex 2 is succinate ubiquinone oxidoreductase, or succinate dehydrogenase. This is a literal TCA enzyme. These are the two separate entry points into the electron transport chain. We do not go from complex 1 to complex 2. Rather, complex 1 and complex 2 both go to coenzyme Q, also known as ubiquinone. Complex 3, or the cytochrome BC1 complex, or the ubiquinol cytochrome C oxidoreductase, transfers electrons from the ubiquinol, which is reduced ubiquinone, onto cytochrome C. Cytochrome C then offloads onto complex 4, which is called the cytochrome C oxidase, and we transfer electrons from cytochrome C to oxygen. Complex 5, which I haven't mentioned until now, is the ATP synthase. This is going to allow the protons to freely flow back into the matrix from the intermembrane space, which will allow the enzyme to synthesize ATP from ADP and inorganic phosphate. So you can see here the oxidation reduction potential steadily drops as we move down to oxygen, which is one of the reasons why, in this case, these enzymes will not run backwards. There's actually an energy barrier due to the nature of redox. The electrons cannot run backwards, they can only run forwards. The ATP, F1FC or ATP synthase, on the other hand, can run backwards because it can use the energy of ATP by converting ATP to ADP and inorganic phosphate in order to pump protons back into the intermembrane space if necessary. So for the complexes 1 through 4, they all have a few things in common. They use electron carriers, including flavoproteins, and these may have flavin mononucleotide or flavonoadenine dinucleotide prosthetic groups, and they will transfer 1 or 2 electrons. We also will see cytochromes, and they're going to contain heme, which is a structure you absolutely must know, and these are going to transfer 1 electron at a time via the iron in heme. Also, we will see iron-sulfur proteins. These transfer 1 electron. Copper in complex 4 is going to transfer 1 electron, but ubiquinone itself can transfer 1 or 2 electrons at a time. Complex 4 is enormous. It's about 40 different peptides. It's 1 megadalton. It is the NADH dehydrogenase, or NADH ubiquinone oxidoreductase, and it transfers 2 electrons, 1 at a time, onto ubiquinone. It uses iron-sulfur clusters. This movement of electrons is going to pump 4 protons. And just briefly, we can see NADH, we lose 2 electrons on the FMN. FMN will transfer 1 electron at a time onto FES, FES then onto ubiquinone. We'll pick up the 2 electrons, once again, 1 at a time onto ubiquinone. Overall pump is 4 protons. Complex 2 is succinate dehydrogenase. And in TCA cycle, what you're going to see is succinate to fumarate, and this transfers 2 electrons to ubiquinone via iron sulfur clusters. However, if you go back and look at FADH2, you'll see it is not a very good reductant. It actually has positive voltage. It's strong enough to move electrons, but this energy is not strong enough to move protons. Complex 3, or cytochrome BC1 complex, cytochrome or ubiquinol, cytochrome C oxidative reductase. This is also pretty big, 11 subunits. We have lots of redox active prosthetic groups. We have hemes, for example, and a Rieske iron sulfur protein, iron sulfur clusters. Now, what are cytochromes? We see these fairly frequently. They have a heme prosthetic group. So the iron changes oxidation state. What's really nice about transition metals is that they act as electron sinks. So they just readily pick up electrons. It's not a problem for them. So if we look at this idea here, we have actual motion from the iron sulfur protein. It will pick up electrons. Okay, and then this is our B position to C position, hence the name BC1. Our iron sulfur protein literally changes conformation to deposit electrons onto the iron of cytochrome C. Okay, cytochrome C is associated with the membrane by electrostatic forces and will accept electrons from complex 3 and move electrons onto complex 4 and donate the electrons at the copper site. Complex 4, cytochrome C oxidase, will remove electrons from cytochrome C, which are going to transfer electrons via copper-containing centers and heme, so copper, iron, onto oxygen. So we're going to need 4 protons to make water, and we also are going to pump 4 protons into the inner membrane space, only 1 electron at a time. We do generate some oxygen radicals, and those could be very dangerous, but they are kept tightly bound to the complex. So here we can see our porphyrin iron structure. So cytochrome C starts depositing electrons, 4 electrons, 4 electrons of iron, copper, iron, and then 4 protons plus the electrons plus the oxygen gives you water. We will also simultaneously pump 4 protons out of the matrix and into the inner membrane space. Okay, we now have that gradient, and we can use that gradient to make ATP. So if you do the theoretical calculations, which I'm not going to ask you to do, 2 electrons from NADH to oxygen will move approximately 10 protons. So here we show the matrix side of the inner membrane and the intermembrane space side of the inner membrane. And we can see that by pumping protons, we have left behind hydroxyl groups, as we split up water. So we not only have a chemical gradient due to the protons, and due to the hydroxides too, by the way, we also have a voltage gradient because we have separation of charge. So this image is flipped from the previous one, but here we can show protons entering into the F0 portion of the ATP synthase, which acts as a water wheel, so the protons move in, rotate around as this whole structure turns, and then are released into the matrix. This energy of this motion is enough to cause the conformational changes which are needed for ADP plus inorganic phosphate to become ATP. And just briefly showing you an electron micrograph that shows that structure. So it's actually been seen, and there is a small video that can be found on YouTube called Inner Life of the Cell Mitochondria, which actually will show you this entire system. It's really neat. I highly recommend you check it out. So while we're here, let's talk about one of the favorites of the old murder mystery tropes, and that is cyanide poisoning, which is usually denoted by the smell of almonds, because almonds actually contain cyanide. So what does cyanide poisoning do? It destroys the electron transport chain, and it does this by binding to the cyanide itself, binds to iron 3 plus in cytochrome C oxidase. And that means the entire electron transport chain shuts down. Because remember, we can't have backflow, so cytochrome C oxidase stops. So we can't put electrons onto water. Well, eventually, we can't offload electrons onto cytochrome C oxidase from cytochrome C, which means cytochrome C can no longer pick up electrons from cytochrome BC1, which means cytochrome BC1 cannot offload its electrons, which means it cannot pick up electrons from ubiquinol, which means ubiquinol can no longer pick up electrons from NADH oxidoreductase. So all proton pumping stops, which means after a bit of time, and it doesn't take much, the F1F0 ATP synthase stops, or maybe even starts to run backwards, because it can no longer use the electrochemical gradient. So the whole system shuts down. So if you ever find yourself in an Agatha Christie murder mystery, I recommend you have on hand maybe some nitrites or thiosulfate. Either one of these will help cure cyanide poisoning. Nitrites will convert oxyhemoglobin to methemoglobin, which will start to pick up the cyanide, pulling it out of the system. And thiosulfates will also directly pick up cyanide to become thiocyanates. You do it fast enough, you just might save the life of a person in a murder mystery. So in summary, the mitochondria is a specialized structure designed for the creation of ATP. We have a series of complexes that carefully move electrons down an oxidation reduction gradient. We do this in multiple steps so that more energy is more efficiently transferred to the production of ATP. If we did it in a single step, we would lose more energy as heat energy, which would reduce the amount of ATP we can make. It is a coupled oxidation reduction series, and it's going to make a very strong electrochemical gradient. We talked about we talked about 5 complexes. And we talked about 4 of them that transfer electrons through iron-sulfur clusters and heme found in cytochromes. We talked about which ones pump protons, basically all of them, but complex 2, complexes 1, 3, and 4 pump protons to the inner membrane space. Complex 5 allows those protons to flow back. And the complex 5 is also the F0F1 ATP synthase, which makes mitochondria. As a side note, mitochondrial diseases are usually maternally inherited because you do get your mitochondria from egg cells. So be sure that you can discuss the entire story of the mitochondria, its structure, its function, the Faraday equation, how it works, why it works, where you could go wrong, how to make sure you're not doing it incorrectly, and as well as the complexes and how they work and how the electrons move through each of the complexes and how protons are pumped as well, why oxygen is our final electron acceptor, what cyanide does, and how to treat it. You want to be able to tell this whole story because these are the types of things you're going to see on the challenge days, and of course these are the types of things that will make sure you have mastery of mitochondrial systems.